I'm going to just say a couple of things before I announce today's speaker. So I'm just coming as the introducer, but also wearing my other hat as the enrichment director for the Elizabeth Caswell, Weiser Caswell Diabetes Institute. So as you may all know, with the new Caswell Institute in our enrichment program, we are supporting a number of speakers across the different departments at the University of Michigan. And that includes a number of speakers in our Caswell Diabetes Institute and MEND Diabetes Grand Round Seminar Series. So you will see a number of folks speaking about diabetes and diabetes-related topics in the MEND Seminar Series over the remainder of the academic calendar year. We already had one speaker earlier this year that was from the Cleveland Clinic. Today is the second in our series, and Holly, as well as folks in the CDI, will be forwarding these announcements around. And I really hope that you will be able to attend all of these speakers. We have a really phenomenal group of external diabetologists giving talks both on the clinical and diabetes research spectra. And then, there will also be opportunities to meet with some of these speakers, and Holly will be reaching out to faculty. But in the event that you're interested in meeting with some of these folks, please get in touch with Holly. So with that, I'm going to introduce today's speaker, Dr. Decio Eizirik, who's the scientific director, the new scientific director of the Diabetes Center at the Indiana Biosciences Research Institute, or IBRI. Some of you may be familiar with this institute because a former member of MEND, Jonathan Flack, who some of you might know, who was working with Martin Myers for a period of time, is new faculty at the IBRI and has started his lab there. And also, Dr. Eizirik is faculty at the Free University of Brussels, and he's coming to us from Belgium today, and a visiting professor at the Danish Diabetes Academy in Copenhagen, Denmark. He's published almost 400 papers and reviews in peer-reviewed journals, well known in the field of diabetes and beta cell biology and type 1 diabetes. A winner of numerous awards, including the JDRF Diabetes Care Research Award, the Albert Renault Prize Lecture in Outstanding Research Achievements in islets from the EASD, as well as the 2013 Rumbaud Award in Type 1 Diabetes Research from JDRF, and serves as an honorary secretary at the EASD, the deputy editor of Diabetologia, and has published absolutely seminal work on the molecular mechanisms of beta cell loss and insulitis and type 1 diabetes, and a real hero of mine as I came up in this kind of evolving idea that is now accepted of the beta cell's pathophysiologic role in the development of type 1 diabetes. So I could say a lot, but I'm going to keep it short so that we can hear from him. And I'll turn it over to him and just say thank you so much for visiting us here at the University of Michigan today.
Thank you very much. This was a beautiful and kind introduction. I am sorry that we cannot be together today. You listed my different positions, but for the time being, I am stuck in Brussels. But let's hope that next year things will improve, and perhaps we will be able to meet in person. So what I would like to discuss with you today is some of our new research lines. As you are going to see, we are trying to expand our work on pancreatic beta cells in type 1 to other tissues in other autoimmune diseases. And the data I am going to present to you today was generated in our lab in Brussels. It's mostly the work of a series of very talented postdoctoral fellows, including Michael Colley, Maria Inês Florian, and so on, both molecular biologists and bioinformaticians. This is my new team in Indianapolis. And there we have a lot of collaborations with both Carmela and Raghu. This is the support, the funding for our research. And we collaborate with people all over the world. A lot of the work on genetics is done together with Lorenzo Pasquali and colleagues and with Fleming Possio. All our proteomics is done together with Thomas Metz and Ernesto from the Pacific Northwest. We also collaborate a lot with Roberto Maloni and Mark Mamula, and all our human islets are provided by Piero Marchetti and colleagues. And in our round center, we collaborate in type 2 diabetes and in monogenic forms of the disease with Miriam Knopp. So as you all know, type 1 diabetes is a chronic autoimmune disease, and we have now a proposal of a new classification, which to my eyes makes sense. So you have individuals at genetic risk for disease, then something happened which still remains to be clarified, could be a viral infection or another danger signal that will trigger an autoimmune assault against pancreatic beta cells. At this stage, the future patients are still normoglycemic, but they start to develop autoantibodies against the pancreatic beta cells. And once the patient has two autoantibodies, you can already call him or her patient because this person has 80% chance to develop type 1 in the coming 10 years. And then there will be a progressive beta cell decline. We don't know how this beta cell decline proceeds because still is not possible to image beta cells in vivo, but eventually there will be a clinical outbreak of the disease. And from there on, the patient will be insulin deficient and will depend on insulin injection for life. And since type 1 diabetes is an autoimmune disease, most of the focus of the research in the field until a few years ago, as Scott said, was on the immune system. And we took a different view. We started to propose the concept that this is not a unilateral disease where the immune system attacks inert beta cells, but a disease where there is a misguided dialogue between the target beta cells and the immune system. What starts this dialogue? It's still an open question. It could be a viral infection or endogenous ligands, for instance, other danger signals. Anyway, these signals will be perceived by receptors that are present at the surface of the beta cells. Perhaps the most important are toll-like receptor 3 and MDA5. And it's interesting that MDA5, which is a receptor for viral double-stranded RNA, is also a candidate gene for type 1. Once this inflammatory process starts, there will be production of cytokines, mostly by local macrophages and perhaps dendritic cells, which will also bind to surface receptors at the beta cell level. And this will lead to a second level of signaling, which is triggering of transcription factors. Perhaps the most important are STAT1 and STAT2, and I am returning to them later, and nuclear factor kappa B. And there are several negative signals here that will downregulate these transcription factors. One of them is another candidate gene for type 1, which is PTPN2, which will dephosphorylate mostly STAT2. The next step downstream of this transcription factor is what we call the effector phase. So up to this stage, we are mostly dealing with innate immune response. Downstream, you are going to start the transition to adaptive immune response, where you have an overexpression of MHC class 1, and this will go together with changes in alternative splicing and endoplasmic reticulum stress, which may contribute to presentation of a modified antigens, both via beta cells that are dying, but also as the CD8 T cells start to come in, via direct presentation in the context of MHC class 1 that may lead to beta cell death. The other mechanism of beta cell death may be secondary to pro-apoptotic signaling and endoplasmic reticulum stress. We need to keep in mind that not all signals induced by cytokines are negative. So, for instance, beta cells will overexpress PD-L1 and HLA-E, which are probably mechanisms to downregulate the invading CD8 cells, but on the negative side, they will produce chemokines that will contribute to further attract immune cells. And they will also upregulate interleukin-17 receptor. So what makes us believe that this dialogue is a real phenomenon? I think one of the things that point in this direction is genetics. So until a few years ago, most people viewed the candidate genes for type 1 diabetes as mostly acting at the immune system level, which is indeed correct. Many or most of these candidate genes act at the immune system level, but not only the immune system level, because when we listed all known candidate regions for type 1, and then based on our RNA sequencing studies of human islets of Langerhans, asked how many of them are present in human islets of Langerhans, we found that 80% of the candidate genes for type 1 are expressed in pancreatic islet cells. And at least half of these expressed candidate genes will change expression when the beta cells are exposed either to pro-inflammatory cytokines or to, for instance, a viral infection. And since we have so many candidate genes that are expressed at the beta cell level, this allows us to do pathway analysis and start to ask ourselves, what are these candidate genes doing in the beta cell? And it's interesting because they are mostly involved in the recognition of viral infection, for instance, via MDA5, and they are also involved in the signaling of type 1 interference. So you have one candidate gene called TYK2, which is a kinase which is immediately downstream of the type 1 interferon receptor, and polymorphisms that lead to decrease in TYK2 expression are protective against type 1 diabetes. And then you have other candidate genes that also downregulate type 1 interferon signaling, including the phosphatase PTPN2, USP18, BAC2, and so on. And you have also just a few, just two actually, candidate genes that regulate beta cell function. One of them is GLIS3, which is perhaps one of the very few who is also a candidate gene for type 2. So these are fundamentally different syndromes, and CTSH. So based on this fact, let's say that many of the candidate genes regulate expression of type 1 interferon signaling at the beta cell level, we grow very interested in further studying the signaling of interferon alpha at the beta cell level. And why is this interesting? First, because of the candidate genes. Second, because when you do histology of the islets of type 1 patients, the most relevant cytokine that you identify in the beta cells is interferon alpha. And the other thing that we have shown a few years ago is that when you treat human islets in vitro with interferon alpha, you will induce production of chemokines, HLA class 1 expression, and endoplasmic reticulum stress, which are again 3 hallmarks of type 1. So if you look at the signaling of type 1 interferons, they will bind to surface receptor of the target cells, they will trigger activation of two kinases, JAK1 and TYK2, which will phosphorylate two transcription factors, STAT1 and STAT2, which will form trimers, either with IRF9 or IRF7, and will go to the nucleus where they will stimulate several interferon alpha beta stimulated genes and will contribute to generate inflammation and antiviral state, and also in some cases favor transition to adaptive immunity. So in order to get a more complete view of the impact of interferon alpha on human beta cells, we have done a time course multi-omic analysis of both human islets and a human cell line, endoc beta 1, exposed to a type 1 interferon. And after 2, 8, and 24 hours, we did in parallel RNA sequencing, proteomics, and ATAC-seq. And ATAC-seq in this context was done to determine areas of open chromatin. And after this was done, this was published a few months ago, we did a detailed data integration. And the first question that you ask when you are doing this type of in vitro work is whether it has any relevance to the human disease. And in this case, we were fortunate that last year, Russell's and colleagues has published a very nice paper where they were able to fax purify, fax sort beta cells from 4 type 1 diabetic patients and 12 controls. And this data is available online, so we could download and reprocess it in our pipeline and compare the genes induced in beta cells from type 1 against the genes induced by interferon alpha treatment. And as you can see, there's a very good similarity, a highly significant similarity between the upregulated genes, but not with the downregulated. And this is probably because we used a short time point, up to 18 hours, when the beta cells are not yet suffering. The next question is whether this is just a consequence of beta cell stress. And I don't think that this is the case, because when you compare human beta cells treated with interferon alpha against the gene signature of islets from type 2 patients, there is no significant correlation. And the same is true when we compare the gene signatures of human islets of Langerhans treated with interleukin-1 beta plus interferon gamma, which will be more relevant at the adaptive immune phase. You have a dramatic correlation of upregulated genes and no significant correlation when you compare with islets from type 2 patients. So this is really a signature of early and late immune assault to the beta cells. So once we have these data sets, we did a protein-protein interaction analysis, and we were helped by the fact that we had both RNA-seq and proteomics, and at least for the upregulated genes, there was a fairly good correlation between expressed genes and proteins. And we start to look, let's say, at the hubs, at the regulatory genes, and we find, for instance, STAT1 and STAT2, IRF7, and here the size of the letters are related to the relevance of the identified pathways. And to our relief, we find interferon signaling, and we also find, for instance, endoplasmic reticulum stress. And one thing that we were puzzling is that we were finding regulatory mechanisms. So these are mechanisms, as I mentioned in the beginning, that probably the beta cell is using to try to downregulate the immune assault. And it's interesting because the beta cell seems to be using some of the mechanisms that tumoral cells use to evade the immune system. Unfortunately, beta cells are much less successful. So what we have found is that the beta cells express PD-L1, and they express it both in vitro, and we also, when we did histology of the pancreas of type 1 patients, you can see that there is no PD-L1 expression whatsoever under basal condition. However, in patients who are affected by type 1 and who still contain insulin-producing cells, you can see that there is a dramatical upregulation of PD-L1 expression. The other gene that we just found is HLA-E. HLA-E is interesting. It's an atypical HLA form who, instead of presenting antigens, has an inhibitory effect on natural killer cells and CD8 T cells. And indeed, when we look at both a human beta cell line and human islets exposed to interferon alpha, there is a clear induction of HLA-E, both at the RNA and at the protein level. And also when we FACS sorted the cells, you can see that HLA-E goes to the surface of the beta cell. And we collaborated with Sarah Richardson and Noel Morgan to do histology of the pancreas of, let's say, normal glycemic individuals and patients affected by type 1, where there are still insulin-containing cells, or patients where all insulin content has been gone. And you can see that in non-diabetic individuals, there is no HLA-E expression. However, in patients with type 1 who still contain insulin, there is a clear-cut upregulation of HLA-E, which is present in both beta and alpha cells, which is slightly different from PD-L1, which is mostly present on the beta cells. When the beta cells are gone, HLA-E disappears. So it seems that HLA-E is another of the potential mechanisms that beta cells use that may delay disease. I believe that this is relevant because we have now in cancer therapy the use of antibodies to block PD-L1 and PD-1, and 1 to 2% of the patients treated with these antibodies develop type 1. And at least 50% of these patients seems to have predisposing genetic risk, and some of them had already autoantibodies. So it may be that expression of PD-L1 helps the beta cell to hang on, at least for a while. And if you remove this defense mechanism, this may lead to a catastrophic outcome, at least in some patients. So we continue with our analysis, and it's interesting because when you look at the attack sec at the chromatin access, there is a very early opening of the chromatin, which is much more marked, is the triple of genes at 2 hours than at 24 hours. And this makes sense because interferon alpha is part of the antiviral response, and antiviral response needs to be fast. So it makes sense that there will be a very fast open of the chromatin. And another thing that was very interesting, and this we saw both for interleukin-1 plus interferon gamma, and this is a study we published together with Quali last year, and the same with interferon alpha. When we start to see the transcription factor sites, let's say, that are bound in the chromatin, we find the usual suspects like interferon response factor, the type 1 interferon classical binding site. But to our surprise, we started to find transcription factors that are involved in the beta cell phenotype, like for instance, NeuroD, PDX1, and FOX. And indeed, when we study the correlation of the binding sites, we find that, for instance, PDX1 co-binds with a transcription, a classical interferon-mediated transcription factor and a binding site, for instance, ISRE. And we studied this further together with Lorenzo Pasquale. And basically, what we are finding when you expose human islets to cytokines is that you have some response elements that we call neo-response elements, where the chromatin is closed. There is no acetylation, because in this case, we also did ship-sack in parallel. And when these cells are exposed to cytokines, there will be opening of the chromatin, and there will be binding of mostly pro-inflammatory transcription factor. However, there is another group of response element that we call it primer. So these are areas where you have already binding of a transcription factor that is involved in the maintenance of the beta cell phenotype, for instance, PDX1. And in this case, the chromatin is already open, and it facilitates the binding and the action of pro-inflammatory transcription factors. And we are studying this further, but this may be one of the mechanisms that explain, let's say, why autoimmune diseases that may have up to 50% similar candidate gene may lead to, for instance, in some cases, rheumatoid arthritis, in other cases, type 1. And when we look at compared the risk variants for type 1 and type 2, you see that in type 1, you find mostly inducible response element, while in type 2, it's mostly stable. And this explains why when you do EQTL, for instance, you find many candidate genes for type 2 in the beta cells. When you are trying to do EQTL for the type 1 candidate genes, you will only detect them when you expose the beta cells to a pro-inflammatory stimuli, for instance, cytokines. And well, we went a little bit further, and perhaps I suggest that you read this review where you get a more detailed information. So I just want to use the last part of the lecture to go to the new data that we are generating now, and which is an attempt to test whether our proposal that there is a dialogue between beta cells and the immune system is specific for the autoimmune disease type 1, or is also present in other autoimmune diseases. So to study this, what we took advantage is of a large wealth of RNA sequencing that has been performed in kidney cells of patients with lupus nephritis, in the optic chiasma of patients affected by multiple sclerosis, and on the joint tissue of patients with rheumatoid arthritis. All this data is available online, so we downloaded it, we reanalyzed all of them through our own pipeline, and once this data were available, the first thing that we did was pathway enrichment analysis, where we compared the pathways that could be detected in beta cells in type 1 against the target tissues of the other 3 autoimmune diseases. And the first thing that you find is that since the 4 diseases are autoimmune, in all of them you find antigen presentation as a major pathway. As you are going to see, there is a lot of similarity in the upregulated pathways, but this is not the case for the downregulated pathways. So the downregulated pathways are mostly disease-specific, and they are related to the tissue function. So for instance, the most downregulated pathway in type 1 is the regulation of gene expression in beta cells, where you find down regulation of several genes like PDX1 and so on. When you look at multiple sclerosis, you have inhibition of protein-protein interaction at synapses. When you look at rheumatoid arthritis, you have inhibition of collagen chain trimerization, and so on. And we continue to look at the upregulated genes, and then basically you start to find interferons. You find type 1 interferons, and you find interferon gamma. And this is present in all autoimmune diseases, even if they are not in the top 20 genes that I am showing here. One critique that we got when we submitted this paper was that the RNA-seq that we were analyzing was bulk RNA-seq, and there was a very minor contamination by immune cells. I would say that the contamination was negligible, but anyway, the referees were insisting, so we managed to download single-cell RNA-seq, again from target tissues of the 4 diseases. And in this case, since when you are doing single cell, you detect much fewer genes, 1 to 3,000 against 20,000 in the bulk tissue RNA-seq, we asked if we could detect interferon stimulated score in the target tissue. And this turned to be correct for all 4 autoimmune diseases. The next thing that we did is a tool called rank hypergeometric overlap. I have already shown a figure because this is the tool that we use to compare interferon-treated human beta cells against beta cells from type 1. So in this case, we used this tool to compare the gene expression in beta cells from type 1 diabetic patients against controls as compared to the optic chiasma in patients affected by multiple sclerosis compared to controls. So here you see that there is a massive similarity in the upregulated genes, and there is very little similarity in the downregulated genes, which is in line with our pathway analysis. And then we compared all 4 diseases together, and you see that when you look at type 1, there is a good similarity of upregulated genes with multiple sclerosis, 1,080 genes in common. With rheumatoid arthritis, you have nearly 500 genes in common. When you look at lupus, you have 624 upregulated genes in common, but you also find 700 genes that are downregulated in one disease and upregulated in the other. And this is mostly related to a neutrophil subpopulation that you have in lupus that apparently you do not have in type 1. The next question that we asked was about the candidate genes. I have told you before that 80% of the candidate genes for type 1 diabetes are expressed at the beta cell level. And we confirmed this in this new analysis. So we asked if this would be true also for the other 3 autoimmune diseases. And indeed, in the 3 of them, the expression of the candidate genes are all above 80%, reaching more than 90% in patients affected by lupus. Again, we needed to deal with the critique if this was not explained by the minor immune cell contamination. And to answer this, we looked at the single cell analysis. And again, we find the candidate genes. And one of the candidate genes is TYK2. And it's interesting because TYK2 appears at a single cell level at the target tissue of the 4 autoimmune diseases. And when we go back to the bulk RNA-seq analysis, you see that you have many genes that are disease-specific, but many genes that are in common at the target tissue level. And there are 4 candidate genes that are in common for the 4 autoimmune diseases, and one of them is our favorite, TYK2. Why is our favorite? Because when we mine our data sets for drugs that may have an opposite effect, and this is a very useful tool that is available, provided by the Broad Institute. So you can find what's called perturbagens. So these are drugs that will induce an opposite effect as compared to the gene signatures that we are finding. So one of the perturbagens that we found was JAK1, and an inhibitor of JAK1, baricitinib, is already being used to treat rheumatoid arthritis, and we indeed showed that baricitinib blocks interferon alpha signaling in pancreatic beta cell, but we were more interested in TYK2 inhibitors as compared to JAK1 inhibitors. And why TYK2 inhibitors? Because it is a candidate gene for the 4 autoimmune diseases. And this has been shown that drug development programs supported by human genetic data are more likely to succeed. And TYK2 inhibitors are already in clinical use for psoriasis. So we managed to obtain 2 specific TYK2 inhibitors from a pharmaceutical company. We are now in the process of testing 3 additional ones from other companies. They are rather specific, so they block TYK2 without affecting JAK1 or JAK3. And basically what we found is that when you expose human beta cells to interferon alpha, you have a massive upregulation of phospho-STAT 1 and phospho-STAT 2, and this can be prevented by both TYK2 inhibitors. And for instance, if you look at HLA class 1 expression, it's not present under basal condition. When you expose human islets to interferon alpha, HLA class 1 goes up, and this is very much inhibited when you expose them in parallel to TYK2 inhibitors. And you also can block HLA class 1 expression, you can block chemokine expression, and also ER stress markers. We also observed that when you expose human beta cells to interferon alpha plus interleukin 1 beta, you induce beta cell death, and this again can be prevented by the TYK2 inhibitor. The other thing that we tested, we were concerned because if you inhibit TYK2 and you inhibit interferon signal, there was the concern that this could sensitize beta cells to a viral infection. But we are not doing a complete blockade of TYK2, it's a 50% blockade. And under these conditions, when we infect these beta cells with Coxsackie virus, you see that you do not increase the expression of the Coxsackie virus protein, or you do not increase beta cell death. So it seems that at least a partial blockade of TYK2 is not going to sensitize human beta cells to infection by potentially diabetogenic virus. So which are the conclusions that we can have at that stage? Some of these conclusions are preliminary. So the first one is that human islets obtained from individuals affected by type 1 diabetes, but not type 2, present a gene signature that is similar to that induced by in vitro exposure to interleukin 1 beta plus interferon gamma or to interferon alpha. And type 1 interferons promotes early changes in chromatin accessibility that are probably necessary for a rapid response during a viral infection. The exposure of beta cells to interferon alpha or to interleukin-1 beta plus interferon gamma activates several cis regulatory elements, many of them presenting enrichment for islet-specific transcription factors. These primary regulatory elements may facilitate cell type-specific responses to pro-inflammatory signals, potentially explaining tissue-specific susceptibility in autoimmune diseases. We find that PD-L1 and HLA class E are checkpoint proteins induced by beta cells during insulitis in type 1 that may at least transitorily inhibit the T cells and nuclear and natural killer cells response. Sorry, I say HLA class E, this is wrong, it's HLA-E. And the second group of conclusions is that there are major common changes in gene expression at the target tissue level in 4 autoimmune diseases, mainly downstream of type 1 and type 2 interferons. 80% of the candidate genes for these 4 autoimmune diseases studied are expressed in their target tissues, suggesting that our previous findings in type 1 diabetes are valid for other autoimmune diseases. We suggested that target tissues should be considered when studying autoimmune diseases to better understand the genetics and the natural history of these diseases. And last but not least, testing the perturbagens detected in the present study may suggest drugs that can be repurposed for the treatment of different autoimmune diseases. And the first candidate here is probably TYK2 inhibitors. This is what I would like to present to you today. And if you have questions, I will be happy to answer.
Oops, sorry.
Go on. I am trying to stop share, but, okay, now. Hello, how are you?
It's Peter. I wanted to ask about the model where I'm imagining that these same cytokines are likely to be important in vivo. Obviously, though, the exposures in vivo are different than we can recreate when we put the cytokines onto the dispersed islet cells. But still, this is a good system to understand responses. I guess my question is, I have 2 very much related questions. One is, if you add the cytokines, and the time course you talked about were things as quick as 2 hours and maybe as slow as 24 hours. But if you add these cytokines and you generate the kinds of signatures you're talking about that include a number of stress responses, is it possible to know if you simply remove the cytokines to terminate the signal, can the cell restore itself back to where it was before you started? And the second question, very much related to that first one. So in vivo, if these are the cytokines that are producing the same kinds of detrimental responses, it is likely that islet cells are seeing them maybe for days, weeks, months, maybe even years. How will we figure out what the kind of response is on a time scale of the real disease? Or do you think that these cytokine exposures in vivo are actually short-lived and the immune system moves on from islet to islet to islet to islet, repeating itself in something that's very much like your in vitro experiment, quick attacks with insulitis jumping, around? Because I'm imagining what it must be like to be an islet in the pancreas of a type 1 patient where this is continuous stuff. It seems like it would be just going on and on and on. And I just wonder even whether the signatures we can see in a day are what we should expect to see in a month and a year, and et cetera?
Yeah, these are two very good questions. The answer to your first question is that if you remove the cytokines, the beta cells recover completely. If you leave them in culture for 3-4 days, they regain function. There are some that are already irreversible damage, they will die. The ones that survive recover. And this is interesting, but we just published a paper together with Miriam Knopp and Piero Marchetti, where we were looking at metabolic stresses instead of, in this case, immunological stresses. And you see that human beta cells, they can recover when you expose them to high glucose, to palmitate alone. However, when you expose to combinations of palmitate and high glucose, their inhibition is preserved at least over a 4-day recovery period. So it seems that the beta cells can recover from some, but not from all stressful situations. Coming to your second question, this is very difficult to answer because the RNA-seq that we have from type 1 patients, they are mostly from people who already got the disease. So it would be wonderful, well, not wonderful, I mean, this would be people who unfortunately die and donate pancreas. But it would be, from a scientific point of view, relevant to have cells from earlier periods. The fact that these cytokines are relevant have been much debated, but 2 weeks ago there was a paper in the New England where for the first time it was shown that blocking one of the cytokines, tumor necrosis factor alpha, is nearly as protective in early type 1 as anti-CD3. So I think this was the first really strong evidence that TNF acts to a large extent like interleukin-1, that they are probably playing a role. So the hypothesis, but this is an hypothesis, that the immune system, as you say, go from islet to islet. They release cytokines in what's called the immune synapse. So they are very close to each other. But cytokines is not the whole story, because once the adaptive immunity kicks in, a lot of the killing is done by CD8, T cells that are killing via Fas ligand and granzyme and perforin. So there are different mechanisms the immune system uses. So my impression at that stage is that these cytokines, particularly interferon alpha, may be important at the early stages of the disease, not only via killing, but via upregulation of HLA class 1, perhaps via induction of ER stress and changes in antigens, in autoantigens, and favoring the move to an adaptive immunity.
Thank you.
You're welcome. Either I was very clear or very obscure. Any other question?
So, quickie, which is what you said the time point you do for these gene signature changes is 24 hours. When you extend the treatment, is the feeling that the gene signature just stays on, just like this?
We have done many years ago, this was still in the microarray time. We did every 2 hours, it was a crazy experiment, in 2 humanized preparations. We went up to 84 hours, if I'm not wrong. And it seems that the bulk of the variation is up to 24. After 24 hours, they stabilize. After 24 hours, you start to see not only pro-inflammatory response like HLA chemokines, but then you start to see suffering. So PDX1 start to go down and so on. So at least between 24 and 84 hours, it's more or less stable. But of course, I would imagine that in vivo, as the beta cells are starting to give up the ghost, you will have other changes that we are not detecting. And indeed, when you look at the comparison between cytokine treated in vitro and human islets from patients with type 1, the clear correlation is with the upregulated genes. The downregulated, we are not peaking to any major extent.
Mm-hmm.
I have a question.
Oh, go ahead.
I was curious, the selection of autoimmune diseases, so rheumatoid arthritis, MS. Did you look at thyroid disease and celiac disease just, because of obviously the high co-prevalence?
Celiac, we didn't look. Thyroid, yes, we just downloaded. There was a RNA-seq of thyroids from patients with Hashimoto thyroiditis. So we are just doing this now. So we don't have as yet data, but this will be the next disease that we're going to look. And we are also getting interested in some degenerative diseases with an inflammatory component. So I want to look at diseases where you have inflammation without much autoimmunity. So this is the direction that we are going down now. Yes, celiac disease would be interesting. And there is a lot of RNA-seq and even proteomics, because people really biopsy the gut. So this is something that we can indeed look. It's a good suggestion.
So can I ask another one? Sorry, Scott, I saw you were.
Yeah, go ahead, Peter. That's fine. I can wait.
His mouth was moving. I caught him like.
That's fine. Go ahead.
Yeah. So obviously, for decades now, a favored model that has kind of fallen into disfavor is the NOD mouse, and there's been quite a bit of transcriptomics done there too. Does this match up? There are those who are proponents of the mouse model as a useful tool to understand type 1 diabetes, and of course there are detractors. Now that you're narrowing down these gene signatures in response to cytokines that match up with human patient data, what is that? Does that allow us to reevaluate whether the NOD mouse captures many of these same genes, including what's it called, the TIC2, and the genes from this JAK-STAT signaling pathway?
It's a good question. We never did this. We have done, but this was more in the microarray time. But there is a lot of new RNA-seq. We didn't do a systematic comparison. We have done histological studies, and you indeed find HLA class 1, CXCL10, and there was a study by colleagues in Yale where they did RNA-seq as the disease progressed. And one thing that was interesting, they found some hardcore surviving beta cells, let's say, that remain after the disease starts, and they are overexpressing PD-L1. And I have this talk with Susan Bonner-Weir because she has this panel of pancreata from golden medalists. These are people who live at 50 years with type 1. I asked her to check if the beta cells, because many of them still have a few beta cells, if they are indeed expressing PD-L1, and I think she's doing this. But I think, yeah, it could be interesting to do a comparison. Even if the signature would be similar, and I guess it would be similar, the problem, for instance, I am more interested at that stage in innate immunity. And the NOD mice is very much geared for a T cell response. I don't think that the innate immunity is so important there. So I think still a useful model, but now that we are starting to get access to more and more human tissue, I rather prefer to stick to human cells. But yeah, I think that's a good suggestion.
And no one has used the perturbogens or the TIC2 inhibitors in the mouse?
They use JAK inhibitors, and we are doing this together with Carmela Evans-Molina, and it seems to work. It seems to work when you do before the disease starts, not after, which makes sense, because this would be a very early phenomenon.
I mean, that's obviously, that is the advantage of the mouse. Exactly what you said earlier, that, you know, you'd like to catch people before the onset of clinical disease, and you really can't. But in the mouse, you really can.
Yeah, no, I think it's a useful model. You need to use it wisely. The way we are working now, we are trying to invert. So we are generating hypotheses in the human tissue, and then we go to the mouse once we have a clear hypothesis, because then the mouse you can study more in detail. So perhaps it's a more rewarding way to go, but time will say.
Thank you.
You are welcome.
I think since you and I are meeting, I'm gonna not ask my question. And since we're at the top of the hour and I know we have a clinical seminar coming up, but just see if there's any other last minute pressing questions. And if there are not, then say thank you very much for a phenomenal lecture. We really appreciated it, the tour de force. And thank you so much for joining us. And I will see you in a different Zoom room.
Thank you very much. It was a pleasure to talk with you, and hopefully we will be able to meet in person. Bye-bye and see you.
We're staying here, though, for this next conference.